Kungliga vetenskapsakademin har beslutat att utdela 2020 års Nobelpris i kemi gemensamt till Emmanuel Charpentier och Jennifer Doudna för utveckling av en metod för genomeditering. The Royal Swedish Academy of Sciences has today decided to award the 2020 Nobel Prize in Chemistry jointly to Emmanuel Charpentier and Jennifer Doudna for the development of a method for genome editing. Die Königliche Schwedische Akademie der Wissenschaften hat heute beschlossen, den Nobelpreis für Chemie des Jahres 2020 gemeinsam an Emmanuel Charpentier und Jennifer Doudna für die Entwicklung einer Methode für die Genomeditierung zu verleihen. L'Académie royale des sciences de Suède a décidé ce jour d'attribuer le prix Nobel de chimie conjointement à Emmanuel Charpentier et Jennifer Doudna pour le développement d'une méthode d'édition du génome. Королевская академия наук Швеции решила сегодня присудить Нобелевскую премию по химии Emmanuel Charpentier и Jennifer Doudna за развитие метода редактирования генома. And you have pictures of the new Nobel laureates on the screen. Emmanuel Charpentier was born in Juvisy-sur-Orge in France. She received her PhD from Institut Pasteur in Paris, and she's currently director of the Max Planck Unit for the Science of Pathogens in Berlin, in Germany. And as many of you know, she made key discoveries while working at Umeå University here in Sweden. Jennifer Doudna was born in Washington, D.C. in the United States. She got her PhD from Harvard Medical School, and she's currently professor at the University of California at Berkeley in the United States. And Dr. Doudna is also an investigator of the Howard Hughes Medical Institute. And as I told you yesterday, the pandemic puts constraints also on the further Nobel Prize celebrations. So we are not requesting that the Nobel laureates come to Stockholm in December to pick up their prizes. Instead, we plan for digital Nobel lectures and a digital Nobel Prize ceremony with laureates participating over video links. We are still working on these events together with the Nobel Foundation and from now on also with the laureates themselves. And we will come back to you with details as soon as possible. But I can guarantee you that Dr. Charpentier and Doudna will receive their awards before the end of the year, and that they will be our guests of honor next time we can celebrate Nobel Week with the traditional festivities here in Stockholm. And with that, I'd like to ask Pernilla Wittungstavsede of the Nobel Committee for Chemistry to make some remarks about the prize. Pernilla.
Thank you, Göran. I'm so honored to introduce this year's Nobel Prize in Chemistry. It's a fantastic prize. Many have been waiting for it, and it's two amazing laureates. To explain their discovery, I will take you from the big scale, things we can see, to the smallest pieces of life that are invisible to our eyes. A human being, like me, is made up of trillions of cells, and thus each cell is tiny, tiny. In each cell, we have our genetic material, the DNA. DNA is long and thin, like a piece of string, that's built up of building blocks called bases. The DNA in each cell contains about 6 billion of bases placed along this string in a particular order. This is what we call the code of life. From this code, thousands of proteins are made in every cell that then perform all kinds of functions in our bodies. So this year's laureates, Emmanuel Charpentier and Jennifer Doudna, have developed an elegant system named CRISPR-Cas9, or simply genetic scissors, that can cut the DNA string at one and only one selected position among all the billions of bases. The ability to cut the DNA where you want has revolutionized the life sciences. We can now easily edit genomes as desired, something that before was hard or even impossible. Today, CRISPR-Cas9 is a common tool in biochemistry and molecular biology labs. It's also used in plant breeding and for novel treatments of human diseases. The genetic scissors were discovered just 8 years ago, but have already benefited humankind greatly. Only imagination sets the limits for what this chemical tool, that's too small to be visible with our eyes, can be used for in the future. Perhaps the dream of curing genetic diseases will come true. Thank you.
Thank you, Pernilla. And now, Claes Gustafsson, could you give us some more insights into the science that led to the Nobel Prize?
Yes. So let's see if we can. So thanks to the development of new chemical tools, we know today the DNA sequences of a large number of different genomes. But to merely read this information is not sufficient to understand life's inner workings. We also need to have tools so we can change the information and find out about the function. With the discovery of the CRISPR-Cas9 genetic scissors, Emmanuel Charpentier and Jennifer Doudna has provided science with such a tool. We can now change the genetic information in any cell and any organism. And we can find out the function of the genetic material. The discovery of CRISPR-Cas9, the system, comes from studies of ancient immune system present in bacteria and other microorganisms. So just as we can be infected by viruses, so can bacteria. And if they suffer a virus infection and they survive that infection, then they keep a piece of the viral DNA as a memory. And this is done in a specific region called CRISPRs, where you have multiple small memory pieces of DNA. So this is where the information is stored. But how can this piece of DNA actually defend the bacteria from later infections? Well, in order to do that, you need to transcribe these regions. So you need to make a copy of CRISPR DNA and make it into RNA instead, CRISPR RNA. And in the next step, you need to actually take this CRISPR RNA, and you need to sort of cut it up in smaller pieces. So you get small pieces of RNA that correspond to one single infection event in the past. And how this processing, as we call it, how this sort of cutting up of the RNA takes place is something that Emmanuel Charpentier demonstrated. She found a new small RNA molecule in the bacteria called tracer RNA. And she showed that this tracer RNA would bind to the long form of CRISPR RNA, and then together with two bacterial proteins, Cas9 and RNase-3, would cut the long RNA molecules up in small pieces. So now there was a piece of RNA corresponding to a previous infection. But how could this RNA piece actually protect against viruses? To answer that question, Emmanuel Charpentier and Jennifer Doudna teamed up. And together the two scientists made a crucial discovery. They found that the tracer RNA that Charpentier found a year earlier, the CRISPR RNA, the small piece of CRISPR RNA, together with a Cas9 protein, formed genetic molecular scissors. And these scissors would use the CRISPR part of the complex and then search for virus DNA within the bacterial cell. And if there was a match where the RNA would match the DNA of the virus, then it would cut the viral DNA and disarm the virus. So this was a great discovery in itself, but the two scientists did not stop there. They wondered, could they make this system even simpler? Because now they had 2 RNA molecules and 1 protein. So what they tried to do was to fuse the 2 RNA molecules into one single RNA molecule, which they call a single guide RNA. And then they investigated if this single RNA, together with the Cas9 protein, could actually target and cleave virus DNA in the test tube, and it could. It worked very nicely. So they had now created a simple two-component system. But what they also did was that they started to make artificial guide RNAs. They changed the sequence of the CRISPR part of the guide RNA. And it turned out they could actually cleave almost any sequence of DNA that they would like to target. They had created a programmable machinery, programmable genetic scissors that could be used to cleave DNA in the test tube. A couple of years later, other scientists, or half a year later, other scientists showed that this was also possible to do in vivo, in cells. And what Charpentier and Doudna had predicted already in the original paper turned out to be correct. This was a system that could be used to cleave DNA in any cellular organism. And why is this possible? Well, and why is this important? Well, it's because if you cleave DNA at the precise site, you can use the cell's own machineries to actually change the sequence there. So if you just cleave the DNA, then the cut will be repaired by the DNA repair system present in the cell. But this repair system is error-prone, so there will be an introduction of errors at the cut. And these errors will inactivate the genetic material there and turn genes off in many of the cases. If you instead would like to change the genetic material in a very specific way, edit it, then you can introduce also a short DNA template, an extra piece of DNA that is similar to the region you want to sort of repair, and that the repair machinery can use as a template. And in this way you can introduce specific changes to any genetic region, genomic region. So what can you use this for? Well, you can use this new technology has transformed the molecular life sciences. We can now edit basically any genome, and we can ask all sorts of different questions. And it can also be used to fix genetic damage. For example, the damage that causes sickle cell anemia. You can use the CRISPR-Cas9 system. You can take out hematopoietic stem cells from a patient, and you can sort of correct for the mutation and then put the cells back. The enormous power of this technology means that we need to use it with great care. But it's equally clear that this is a technology, a method that will provide humankind with great opportunities.
Thank you, Claes. Great discovery and a great prize. And now it's time for questions from you. We are hoping to get one of our Nobel laureates with us on a phone line, but it's not ready yet. So in the meantime, perhaps you have some questions to us in the panel. And remember to push the button on the microphone stand and make sure the red light is on if you're going to ask a question. We had some problems with that yesterday. Who would like to start? Well, if you don't ask now, you won't get the chance later. Yeah, Maria Gunther, please.
Yes, this is Maria Gunther from Dagens Nyheter. Did you ever consider to include anyone else in the prize?
I'll give that question to Claes.
So this is a question we never answer. We are just extremely happy for this year's laureates. It's a big field, and there's a lot of good science being done in this field. But we have decided this year to award the prize to Charpentier and Doudna, and I can only say that.
Yes, David Keaton.
Thank you, David Keaton, Associated Press. You mentioned earlier that that imagination was the only limit to what this prize and the science behind this prize can achieve. Is imagination the only limit, or is ethics also an important component?
Pernilla?
Yeah, that's a good point, and of course you're right. Ethics, laws and regulations are extremely important here as well, and exist already in place and are being developed, as Claes was alluding to. So, yes, I was trying to be brief.
I could add to that there's recently been a commission appointed by the National Academy of Sciences in the United States and the Royal Society of Britain, and in which one of our Academy's members, Anna Videll, also took part, that has presented some guidelines for use of this kind of genetic technology. So there is a lot of work going on, and criteria are being developed by the scientific community. Now I think we may have Dr. Emmanuelle Charpentier with us on the phone line. Are you there?
Yes, I'm here.
Very good. Thank you for being with us at the press conference. We're in the midst of it right now. Who would like to ask a question? Yes, we have a lady over there.
Thank you, Chairman. I'm a journalist from Nordic Chinese Times, and congratulations to this year's winners. People found that many great scientists, including 70% of the Nobel Prize winners, believe in theology. When they face limitation, they pray to God for wisdom. Do you believe in theology? Do you, in addition to doing research every day, do you talk to God? Can you combine chemistry and theology to do contribution in science?
Okay, that was a very private question. You answer it if you feel like it.
I'm not sure whether I understood it. The question is whether I'm talking to God.
Yeah.
No? I'm obviously, obviously maybe people don't know it, but it's not a secret. I was raised Catholic. Now my religion turned out to be quite much science, in the sense that all my focus is on science, and I believe in what I'm doing as a scientist, and what science shows us in terms of at least how do you say showing us the nice part of nature, which is what the CRISPR discovery has shown. Very nice and sophisticated, yet simple mechanism of life that can be used for helping society and humanity.
Thank you.
Maybe it does not answer directly your question, but okay.
It was an elegant elaboration. Thank you. Now, Thomas von Heine.
Congratulations. This is Swedish Television Public Service. We know that we've been waiting for this particular prize for some years now. Have you been waiting for it, expecting it?
Actually, what was maybe I should say not common or quite unique is that very fast after the publication of the Nature paper in 2011 and the Science paper in 2012, it was indeed mentioned to me a number of times, maybe more than what I would have liked, that one day, this so-called discovery may be awarded the Nobel Prize. I have to say that, it's something you hear, and as much as I have been awarded a number of prizes, it's something you hear, but you don't completely connect. when Göran Hansson called me this morning, I have to say that then I realized that, yeah, I was very emotional, I have to say, because I was, strangely enough, I was told a number of times, but when it happens, you're very surprised, and you feel that it's not real. But obviously it's real, so I have to get used to it now.
Thank you. Yes, please.
Anneli Megner-Arn, Swedish TV4. Congratulations on this prize. Yes, we waited such a long time for it. I wonder if you can tell us a little bit about what it was like at the Umeå University. I've heard that you stayed up late at night to talk to Jennifer and how you did this together. So could you tell us a little bit about how it was working there?
Yeah, Umeå was a great time in my career. I will try to not be too long, but at first when I announced to my colleagues in Vienna and to my friends and family that I would accept a position of associate professor at the Laboratory for Molecular Infection Medicine Sweden. That is a Nordic partnership together with EMBL for molecular medicine at Umeå University. The people who knew me very well wondered whether I was clear in my mind, because they were thinking that they were very surprised that I was taking such a decision. As a matter of fact, I was supposed to go to the Karolinska Institute right after my postdoctoral time in the US. This was in 2002 in the lab of Stefan Normark. Ultimately, I went to Vienna, so I always had in mind that one day I absolutely needed to go to Sweden, and in 2007, the means was advertising positions, and I thought that it was exactly what I needed at the time of my career, even though for sure it was at maybe a certain price for my personal life. But once in Umeå, I felt so comfortable. I say this because it's part of it. With very nice colleagues, very nice environment, a supportive environment, a freedom to do research, a sense of quality versus the time and the pressure to just publish for publishing, but the sense of giving the scientists and even the young scientists the recognition of the ideas they may have, giving them the chance to pursue what they want to pursue, even though it may be not that clear at first or excited at first. And this, it was very, very important for me. This is the reason also why I took the decision to go to Umeå, because I felt that there would be a community understanding what I wanted to do. And the research really started already in 2007. We had identified tracer RNA in my former laboratory at the Max Peretz Labs at the University of Vienna in Austria. And I had my interview in 2007, and I was starting to go back and forth in 2008 to Umeå, and this was really during this traveling that I really decided to actually really focus on CRISPR, which is the research that we had already started in my lab. And then it was in a way a long way to a lot of work and commitment, but results that were so clear thanks to the nature of the CRISPR-Cas9 system in Streptococcus pyogenes and other bacterial species. So this was really a very unique time for me also. I've run a lot of projects myself, a lot of projects are running in my lab. This was really specific. With regard to Umeå after, when you live in Umeå, you know that the nights are very long in the winter, and it's very cold, and in the summer the nights are very short. There is even not really night. And at the time when we were writing and finalizing the manuscript together with Jennifer Doudna and the team and Martin Linek and Christoph Schilinski, it was good for me because I was up the entire day and the entire night. I will be leaving at the Umeå time, and I will also be leaving at the Californian time. So indeed, I will go back to my home at 3 a.m. in the morning, full light, get a few hours of sleep, and go back to the lab.
Well, thank you. I hope people in Umeå heard that. I'm sure they're going to love you even more after that. Who would like now? Yeah, I think Maria Gunther was first.
Yes, this is Maria Gunther from Dagens Nyheter. Congratulations to the prize. I think we are almost as happy as you are. When did you first realize what this tool could be used for?
I think this actually, it has always been part of my, if you wish, of my education. So I started as a bacterial geneticist. I started to manipulate DNA in bacteria for the purpose of engineering bacteria genetically already at the start of my career at the Pasteur Institute, and then at the Rockefeller University and NYU Medical Center, where actually I developed a genetic toolbox for Staphylococcus aureus and other gram-positive bacteria. During one of my postdocs at the New York University Medical Center and the lab of Pam Cohen, I was also focusing on engineering transgenic mice to understand proteins that are involved in cell-cell adhesion and potentially have a role in skin diseases, because I was interested in understanding skin in eukaryotic systems to be able after to pursue my research on infectious diseases and thereby choosing Streptococcus pyogenes, that is a skin pathogen. At the time, it was at the end of the 90s, it was clear that to perform knockout, one needed to go through mice, and that later on the RNA interference was a way to affect the expression of genes in human cells, and after Zinc-finger nucleases, Thal nucleases came. But for me, I was always having in mind and observed that one day I would be very happy if one could really perform proper genetics. That was always my, let's say, my leitmotif for the bacterial world, but to be able also to do proper genetics in human cells and thereby develop, let's say, providing the scientist means to develop models of diseases in human cells that are more relevant to the clinical situation, and also provide tools that would allow them to understand better as well human diseases. Having said this, for sure, working on the CRISPR RNA was also within the frame of the idea of working with small RNAs in bacteria, whereby this type of research, if you wish, would always lead potentially to either a way to discover new pathways that could be useful for developing therapeutics against bacteria, but also a way to find new mechanisms to target genes and their expression. You know that RNA interference are also small RNAs, and a lot of molecular biology tools in any case originate from discoveries of enzymes or associated elements in bacteria. So this was always in my mind, and was always a leitmotif as well to work on CRISPR. And quite early on, actually, I think quite early on also for CRISPR, I do wish that it could be also harnessed to treat human genetic disorders.
So a vision that was there early on and then matured and modified over time. There, yes. A question here. We have a couple, I think we have time for two, maybe three short questions and answers before we have to conclude. Please.
Thank you, David Keaton, Associated Press. First of all, what potential applications excite you the most today when we look at the applications of this discovery, and specifically looking at new vaccines, potentially for COVID, looking at the developments that are made in DNA vaccines? Are you hoping, or do you expect to see this research feed into applications that could contribute to solving the corona crisis that the world is facing today?
So, first of all, maybe you have seen that there are some kits based on the CRISPR-Cas system and that are available to be able to detect the virus. Having said this, CRISPR has a direct technology to cure, to at least, with regard to really being developed as a vaccine. I don't see it in a direct way, but in an indirect way, yes. We are performing CRISPR screens in our lab with human cells infected with SARS-CoV-2. Others are doing it as well for the hope to be able to find new components or new molecules important for the virus to replicate and cause diseases. And these findings could help to develop a vaccine. So I see it more with regard to research and development rather than the CRISPR technology itself being developed as a vaccine.
Thank you. Do we have, no? I think there was a gentleman here.
Yes, congratulations also from me, Steffen Trumpf from the German news agency DPA. I think that you and Ms. Doudna are the first woman sharing the prize. Therefore, my follow-up question, how excited are you about that point of your prize?
I tend to consider myself as a scientist, but surely I'm very happy that, as a matter of fact, for this prize, these are two women. I wish that this will provide a positive message specifically to the young fellows, the young girls who would like to follow the path of science, and I think to show them that in principle women in science can also be awarded prizes, but more importantly that women in science can also have an impact through the research that they are performing.
Indeed.
I think it's very important nowadays, but not only for women. We see, let's say, a lack of interest of following the scientific path, which is maybe worrying. So I wish that the fact that Jennifer Doudna and myself are awarded the prize today can really provide a very strong message for the young girls.
Thank you. Thank you very much. And thanks for being with us today on the phone line at the press conference, Manuel. And we'll be in touch about the further arrangements. Congratulations again from all of us in Stockholm. Congratulations to your prize. Bye-bye.
Thank you very much. Bye.
Are there any further questions? If not, we'll conclude this press conference. There will be interviews in various rooms. You will be shown how to find your way to your interviewee, and be careful and keep the distance when you exit the room. Thank you very much.